Welcome to the second series of perception, the vision. Most of our impressions of the world and our memories of it are based on sight. Most of the time, we take a photo with a camera to keep a nice experience from our vision. But they're saying, keep it with your eyes, not with photos. That is true. Our vision and camera give different properties of the scene. A camera reproduces point by point the light intensities in one plane of the visual field. However, unlike a camera, the visual system can create a three-dimensional representation of the world from the two-dimensional images on the retina. Also, the brain analyzes scenes into distinct components by separating the object from the background to determine which light stimuli belong to which object. During this process, our brain uses experience about the structure of the world. In this video, we lay a foundation for understanding the neural circuitry and organizational principles of the visual pathways. Also, I would like to address the introduction of the neurosignal of visual processing. Okay, let's briefly talk about how the brain processes visual information, not only the properties of the stimulus, but also on its context. The brain analyzes visual scenes at three levels: low, intermediate, and high level. At the low level, the visual scenes is discriminated into local contrast, orientation, color, and movement. The intermediate level involves analysis of the layout of the scenes and surface properties, tracing the visual image into surfaces and global contours, and distinguish foreground from the background. The highest level involves object recognition. Once a scene has been traced by the brain and objects recognized, the objects can be matched with memories of the shape and their associated meanings. Vision also has an important role in guiding body movement, particularly hand movement. Visual processing begins into two retinae. The axon of the retinal ganglion cells are the projection neurons of the retina. They form the optic nerve that extends to a middle line crossing point, the optic chiasm. Beyond the optic chiasm, the axons from nasal and temporal hemiretinae carrying input from one hemifield join in the optic tract, and the optic tract extends to the lateral geniculate nucleus of the thalamus. Therefore, the primary visual pathway is also called the geniculosrate pathway. The thalamic neurons then relay retinal information to the primary visual cortex. Secondary visual pathway from the retina runs to the superior colliculus and is important in controlling eye movements. This pathway continues to the pontine formation in the brainstem and then to the extraocular motor nuclei. The third pathway extends from the retina to the pretectal area of the midbrain, where neurons mediate the pupillary re-axis that control the amount of the light entering the eyes. The object features, pattern, color, motion, and depth are perceived in the discrete areas of the cerebral cortex. The visual areas for the cortex can be differentiated either by their representation of visual space, known as visual stopic, or retinotopic map, or by the functional properties of their neurons. Functional specificity has been demonstrated for a number of these areas. Ventral occipital 1 is implicated in color processing. The lateral occipital complex, called object shape, fusiform face area, called faces. The parahippocampal place area responds more strongly to the place than to objects. The extrastriate body area responds to the body parts than object, and visual area 5, or middle temporal area, is involved in motion processing. Areas in the intraparietal sclus are involved in the control of spatial attention, saccadic eye movement. There is a model of neural processing of vision which is called the two streams hypothesis. The dorsal pathway courses through the parietal cortex, a region that uses visual information to direct the movement of the eyes and limbs, that is, for visual motor integration. The ventral pathway extends into the temporal lobe. The inferior temporal cortex stores information about shape and identities of objects. These were the introduction of the pathways of the visual processing. Let's move on to the important concept to understand how neural processing works in the visual system. Our capacity for understanding both the object and the surrounding scenes, the detail and the context, give us a powerful ability to interact with the world. Neurons can achieve this in part because their receptive field can extract both local and global features. A field is a group of objects or properties that share the common characteristics. Similarly, the receptive field of the neuron is the portion of the sensory field that affects the signaling of the neuron. For example, the entire area that an eye can see is called visual field, and the patch of the visual field that any single neuron monitors are that called receptive field. We will dig in more details receptive fields in the next video. The dominant feature of the functional organization of the primary visual cortex is the visuotopic organization of its cells. It means that the visual field is systematically represented across the surface of the cortex. In addition, cells in the primary visual cortex with similar functional properties are located close together in columns that extend from the cortical surface to the white matter. The properties that are developed in the primary visual cortex include orientation, specificity, and the integration of the input from the two eyes, which is measured as the relative strength of input from each eye, or oscular dominance. Oscular dominance columns reflect a segregation of thalamocortical inputs arriving from different layers of the lateral geniculate neurons. The columnar system serves as the substrate for two fundamental types of the connectivity along the visual pathway. Serial processing occurs successive connection between cortical areas, connection that run from the back of the brain to the forward. At the same time, parallel processing occurs simultaneously, subsets of fibers that process different submodalities such as form, color, and movement. Many areas or visual cortex reflects this arrangement. For example, functionally specific cells in V1 communicate with cells of the same specificity in V2. These pathways are not segregated. However, there is some mixing of information between different visual attributes. Each area of the visual cortex transforms information gathered by the eyes and proceed at earlier synaptic relays into a signal that represents the visual scene. The principal input to the primary visual cortex comes from two parallel pathways. These pathways originate in the parvocellular and magnocellular layers of the lateral geniculate nucleus. This characterization of parallel pathway is only an approximation, as there is considerable interaction between the pathways. This interaction is how various visual features, color, form, depth, and movement are linked, leading to an unified visual perception. There is another important concept to understand how visual information is processed: neural code. Individual neurons in a sensory pathway respond to a range of stimulus value. For example, a neuron in color detection pathway is not limited to responding to one wavelength, but is responding to a range of wavelengths. A single visual percept is the production of the activity of several neurons operating a specific combinational and interactive fashion called a population code. The population code is that visual content is represented at each stage by a pattern of activity across the local population of neurons. Population coding has been modeled in various ways. The most prevalent model is called vector averaging. We can illustrate population coding with a population of the orientation selective cell. Each of them responds optimally to a line with a specific orientation. Each neuron responds not just to a preferred stimulus, but rather to any line that falls within a range of orientations described by a Gaussian tuning curve with a particular bandwidth. A stimulus of particular orientation most strongly activates cells with tuning curves centered at the orientation. Cells with tuning curves centered away but overlapping that orientation is excited less strongly. The cell signals can thus be represented by a vector pointing in a direction of the cell's preferred orientation with a length proportional to the strength of the cell's response. For all the activated cells, one can calculate a vector sum with a direction that represents the value of the stimulus. Thank you for watching. That's all for the introduction of the vision series. Next video will be the low-level processing, the first part of the vision series. See you then.